Good afternoon, everyone. We're going to go ahead and get started. My name is Kamala Visweswaran. I teach in the Ethnic Studies Department here at UC San Diego. And this event is being put together by the South Asia Initiative, which is a group of faculty from across campus that is trying to organize a series of events that bring together people from the humanities, social sciences, and STEM fields to share perspectives about urgent medical, social, and political issues. Last year, we did an event on climate change. So South Asia is actually our focus, and we do a number of events that are roundtables like this one today. And we also show documentary films. We hold talks and lectures from literature, history, political science, economics, medicine, and so on. Next week, we'd like to invite you to attend an event by a Kashmiri documentary filmmaker, Ifat Fatima, who will be speaking at this same time, 3:30 to 5 p.m. in SSB 107. That should be of great interest. If you're interested in joining the South Asia Initiative, we have faculty from across 9 different divisions and schools. We're quite interdivisional and interdisciplinary. We have 6 faculty members who currently serve on our steering committee. Myself, Prashant Bharadwaj in economics, Lily Irani from communications, Saiba Varma from anthropology, Steve Parrish from anthropology, Vish Krishnan from the Rady School, and Vineet Bafna from CSE, to whom I will now turn over the rest of the introductions. Thanks again for coming.
Thank you all for coming. As Kamala said, we are definitely excited to host events pertinent to issues of South Asia, and of course malaria being endemic in South Asia. It's my pleasure to introduce a really distinguished panel today of people who are doing some of the most cutting edge work in both in genetics and also in sort of drug screening. So Ethan Beir holds the Tata Chancellor's Endowed Professorship in Cell and Developmental Biology at UC San Diego. After his PhD from Harvard, his research is focused on using the model organism Drosophila, the fruit fly, as a model system both in studying developmental processes, specifically patterning in Drosophila, and also using it as a model to understand diseases. And in recent very exciting work, which he's going to talk about today, he's engineered new mechanisms for active genetics which allow organisms to bypass traditional laws of Mendelian inheritance, and you can use these in applications for sort of gene drives in mosquitoes and so on. He has received numerous awards for his research, including the Sloan Foundation Award, the McKnight Award for Neuroscience, the Basil O'Connor Award, and the American Cancer Society Junior Faculty Award. We also have Professor Elizabeth Winzeler. She's a professor of pharmacology and drug discovery in the Department of Pediatrics here at UC San Diego. And she leads a group that uses systematic, data-intensive methods to solve problems at the interface of host pathogen biology, typically involving large screening methods. And her recent work has focused on identifying mechanisms of drug resistance and also discovery of new gene targets for the malarial parasite. Dr. Winzeler is a fellow of the American Academy in Microbiology and has received, again, numerous awards from the Keck Foundation, the Ellison Medical Foundation, and the Bill and Melinda Gates Foundation. In 2014, she received the Bailey Ashford Medal for Distinguished Achievements in Tropical Medicine. She received in 2017 the Medicines of Malaria Venture Project of the Year Award and was just awarded the 2018 Ellis and C.C. Wong Award in Molecular Parasitology. And then last but not least, we have Professor Karthik Muralidharan from economics, where he holds the Tata Chancellor's Endowed Chair in Economics. Born and raised in India, he earned a bachelor's from Harvard, an MPhil from Cambridge in England, and also a PhD from Harvard. He's a research associate at the National Bureau of Economic Research and a fellow and board member of the Bureau of Research and Economic Analysis of Development, in addition to many other affiliations. His primary research interests include development, public and labor economics, and specific topics of interest include education, health, social protection, measuring quality of public service, and improving the effectiveness of public spending. So without further ado, I'm going to ask each of the panelists to come and make a short presentation at first, and then we'll invite all of the panelists on stage to address any questions. So Ethan, we'll start with you. Welcome. And let's give them a couple.
Thanks. Thank you very much. So appreciate the invitation to contribute to this, participate in this interesting event. And so I'll just give a quick overview of what it is that has been done in a collaborative effort between my group and the group of Tony James at UC Irvine in the pursuit of the use of gene drives as a tool to combat malaria. And this was all initiated by work that started with Valentino Gantz, who was a graduate student at the lab when he came up with this idea of essentially turning the fruit fly into a little PCR machine, the polymerase chain reaction here, to create a genetic equivalent of it that would spread a genetic trait transgenerationally in a very efficient manner. And the basis of it comes from a technology which I'm sure you've all heard of, and I'll try to go through it pretty quickly just to give you a sense of how it works, but it's got enormous transformative applications and implications for modern molecular genetics, called the CRISPR-Cas technology. And what it is basically is it's a two-component system that consists of a small RNA, which is this green moiety here, that binds to a blue protein called Cas9 that when it binds the guide RNA, this RNA that's called a guide RNA or a gRNA, when it binds it, that guide RNA brings this protein, which is a kind of a molecular scissors that can cut DNA across both strands, it brings it to a particular sequence in the genome. And the big thing about this is it's totally programmable. So this piece here you can have made to be anything you want, and that takes you pretty much to anywhere in the genome, and you can cut the genome anywhere you like. And that is what is so transformational about this technology. It can be used in a myriad of ways to cut the DNA and then, according to the sort of vernacular, edit it by following up on that cut, having the cut repaired in such a way that a desired change gets made as a consequence. And the way it works is kind of like this. All of us inherit one chromosome from mom and one chromosome from dad. We can call them A and B. And you can imagine that the Cas9, once bound to the guide RNA that cuts in a particular place where it's programmed to cut, cuts one of the chromosomes, A in this case, in that position. And it cuts both strands. And that basically separates the chromosome into two parts, which means that a cell that has that will die if it doesn't fix it pronto. So there are two different pathways that the cell has, and this is a gross oversimplification, but nonetheless, there are two general pathways by which this can be repaired. One is considered to be an error-prone pathway that has this funny name here that is kind of abbreviated NHEJ. And you can think of it as if you had a big, there's like a rope got cut and you just wanted to do anything to stitch it back together. You maybe tie the ends very crudely to each other just to get it back together. But it's not exactly a nice little rope. There's a knot, an ugliness in there. So there's a change in the sequence. So it's a mistake right at the place, but it puts it back together so at least the cell can live. And then there's a more elegant way of doing it, which is to simply copy information from the chromosome that's still intact into the hole and then fill it with the correct sequence, at least the correct sequence as it is present on the B chromosome. It can be a little bit different sometimes between A and B, but essentially the information would be intact information that goes into the cut. And fortunately for the idea that Valentino had in mind, this is the pathway that predominates in the cells that give rise to the germline, the reproductive cells, the eggs and the sperm, which is a good fortune for Valentino, because whenever this happens, that process that I'll just describe in a moment would not take place. But fortunately, this is the overwhelmingly used pathway in the germline. And so what Valentino thought is he thought, well, maybe I can trick the genome into accepting a piece of DNA into it by making a kind of fake little chromosome. And so what he did is he took the Cas9 and the guide RNA and he put them on a little piece of DNA, and then next to them he put little pieces of the genome that were immediately adjacent to the place where these were programmed to cut. So the idea is then these come together, the Cas9 and the guide RNA come together because they're produced from this piece of DNA that gets injected into the cells that are going to make the germ cells of the organism. And then it cuts the DNA of the, in this case, the fruit fly, that's the one we started off with, but then will be the mosquito, cuts the DNA at a specific location. And then this piece of DNA corresponds exactly to this one. This piece of DNA corresponds exactly to that one. So what the repair system thinks is that, well, maybe this is a little fly chromosome, so it comes in here and it sees a cut and it copies then the information from what it thinks is the other chromosome into the gap. And so now the genome has the Cas9 guide RNA molecular scissors inserted into the genome precisely at the point where they're programmed to cut it. And the effect of that is that in the next step, what happens is that the Cas9 and the guide RNA get made from being inside the genome, so now it's part of the organism itself, the fly, and it cuts the opposite chromosome, because remember there's two, one from mom, one from dad. It cuts the other one, and now the gap here, in the same way, gets repaired by copying information from the other intact one. That's what happens in the germ cells. And so now the consequence is that both mom's copy and dad's copy have the genetic element, and all the kids then inherit it because there isn't any chromosome that doesn't have it that can be inherited. So by force, then they, both mom and dad, but both of these chromosomes are passed down to, one or the other of these chromosomes are passed down to the offspring, but both of them have the sort of genetic widget inserted into it. And so this is a little illustration and animation that Valentino came up with. Flies start off being brown in body color, and he created this genetic widget that would go into a gene that's necessary for making that brown color, and when mutated, makes a fly yellow. And so his element cut like this and copied over to the other chromosome, and then was inherited by almost all of the offspring. And so that was kind of the genesis of the idea of using this system as a way to potentially bring into the genome a genetic element that all the offspring would inherit. And if all the offspring inherit it, then there are ways of using that to spread a desired genetic trait into populations. And we were unaware of this at the time that we were doing these experiments, but this was kind of the holy grail among some in the field who were thinking about ways of eradicating malaria, like Tony James, who I'll get to in just a moment. And so again, the basic idea here is if you imagine a normal situation that you might have learned about in your high school genetics, if there's a characteristic like the light body color compared to the normal body color of the fly, and it's the formation of pigment, so that if you inherit, as is typical from one parent, this copy that doesn't make the pigment, and from this parent one that does make the pigment, then the kids all have one copy that makes the pigment, one that doesn't. But since making pigment is dominant, it's the presence of the pigment, then all of the offspring are wild type in appearance, normal in appearance. So you have a 50% inheritance of the trait that comes from this mutated form of the pigmentation gene, and 100% of the progeny at this first generation look normal. And so with Valentino's genetic widget, the same thing happens initially, where you get one copy of one chromosome from one parent, one from the other, so they've got one of each. But then the difference is that the red one here, the mutant one, converts the other to its same state. And so now you have both chromosomes that that have the characteristic 100% inheritance of the trait and 100% mutant. So it's a very different thing. It's as though a blonde person, say, married a dark-haired person, and all of their kids were blonde instead of dark, which is the typical thing. And not only that, but all their grandkids were and their great-grandkids. So it's a kind of different form of inheritance. And so in mosquitoes, the idea was the immediate possible application was in spreading a trait that would be linked to that genetic device that could be a beneficial one for combating malaria in one way or another. And so the idea is that if green here is the element that copies, if this one mates with a wild type, a normal mosquito, then all of its kids will be green. And then they mate with other wild type mosquitoes, all their kids are green. And then they mate with wild-type mosquitoes, and then all of their kids are green. And eventually, as this process goes on, the entire population comes to carry the genetic element. And that process is called gene drive, the ability for something to be inherited more than just by chance, that is 50% of the time. And so if you look at this in terms of the issue of the problem of malaria, and of course we'll hear a lot more in an expert fashion from Elizabeth in a moment about this, but it's endemic to many of the tropical areas of the world, and we are particularly focused here at UCSD on India because of a very generous gift that was given to the university by the Tata Trusts that is very interested in trying to use this type of technology in the context of India and, of course, then potentially more broadly in South Asia. But and malaria worldwide has a huge burden and is perhaps killing still at this time, even though there's been quite a bit of progress in combating it, as much as 500,000 people per year are fated to die from it. And there are many ways that are currently being used to combat malaria. There are insecticide impregnated bed nets that have made a huge impact. Indoor residual spraying, which is to make sure that mosquitoes that touch a wall, which they will if they feed on you and then fly off to the nearest wall to hang out for a while to digest their evil meal, then they would contact the pesticide and die. Of course, sanitation, just getting rid of the places where they breed. And as Elizabeth will tell us much more about, of course, a very important component are drugs that humans can take that can help cure or prevent infection by malaria. But in any case, with respect to our strategy, the idea is that you imagine a mosquito, an uninfected mosquito, bites a person that's infected that carries the malarial parasite. Then the mosquito gets the parasite. It becomes then a carrier of those infected agents, those parasites, those plasmodial parasites. And then it flies over to a naive, otherwise happy human being, and then transmits those parasites to them to make them diseased. So there are two general strategies that people have thought about in terms of using gene drives, which can disperse genetic information throughout a population to help with this problem. The first one is to use gene drives in what's known as suppression, which is basically to kill the mosquito. So in this kind of scenario, people design a genetic element that will just make the mosquito go away. So it either kills it or sterilizes it. And there's a whole group of people at Imperial College in London, funded heavily by the Gates folks, that are trying to do that. And the alternative strategy, which is the one that we favor, and I'll discuss why in a moment, is one that we refer to as immunizing, or replacing the wild-type population of mosquitoes with ones that are no longer capable of transmitting the disease. So we immunize the mosquito against being able to propagate the parasite, so that now the mosquito bites another person, but it doesn't transmit the parasite, so that doesn't spread the disease. So what are the merits of these killing or suppression versus immunizing or modification strategies? Well, if you do suppression, then you drive, again, a lethal or sterile characteristic into a population so that the population collapses. And if you do that effectively, then it reduces all of the diseases that could be carried by a given mosquito. So, for example, some mosquitoes, like the Aedes mosquitoes that carry viral diseases, carry dengue, chikungunya, and Zika and yellow fever, all the same mosquito. So if you get rid of the mosquito, you get rid of all of those diseases at the same time. So that's a plus for sure. The weakness, though, is that it's not particularly stable in an area where the mosquito is endemic and where it's deeply spread throughout all of the ecologies, because what you'll do, it's basically a genetic insecticide. What you'll do is you'll wipe it out, if it's effective, throughout much of its range, but there'll be a few isolated pockets where you don't get to it, and because it's built to destroy itself at the end, because the mosquitoes that have it, they die or they don't reproduce, so they go away. Then those that haven't encountered that, haven't mated with those, then they are okay, and there's no more of that genetic element around in the environment anymore, so they come back, and then you have to do it again. So it means that you keep having to retreat the same area over and over again. The idea that it could potentially do ecological harm by wiping out a mosquito, that's something to consider. I personally don't think it's a very large threat, but it's something that one needs to consider nonetheless. And in other situations where it's not a single mosquito in the ecology of vast numbers of different species of mosquitoes, but if it was a more rare organism that was a keystone organism in a food chain, then you'd have to be careful about that particular element. But I think with mosquitoes, it's probably not much of a concern. But in any case, the immunization strategy uses the drive to modify the organism. And again, we'll talk about that more in a moment. But the idea is just to prevent the parasite from being transmitted in this case. And it's stable since it doesn't reduce the mosquito populations. The idea is to insert one of these genetic elements in a region of the genome that's silent between genes. So it doesn't harm the mosquito in any way. It just silently spreads through the mosquito and just makes it so that the mosquito can no longer propagate the parasite. So since there's no evolutionary selection against the mosquitoes that carry that, and they can perdure in the environment for a long time, they'll eventually get to those little pockets that would otherwise be missed by one that tried to suppress the population. And not only that, but mosquitoes that then fly in from outside will then mate with those that have it, and it'll spread to those. So it's very stable. And it does the least ecological harm possible if you're worried about that, in the sense that you aren't hurting the mosquito. I mean, there's presumably not much of an ecological value of the parasite itself. The weakness is that you have to then make an immunization element that is useful for each disease. And that, of course, if there are several different diseases that are carried by that could be something that takes some effort. Although, in principle, if you figured out how to do it for one, it may not be such a big increment to then do it for another. Now that then brings me to our close collaborator and buddy, Tony James, up at UC Irvine, who has spent his entire career, like Elizabeth has, trying to combat malaria. And so Tony was a leader in creating the first genetic tools in mosquitoes, molecular genetic tools, transgenesis. And then he spent over a decade building a very complex genetic device, which is depicted here in a highly simplified form, that consists of two important components, one of which is a piece of DNA that you can think of as a switch that turns the gene on or off, but it only turns it on after females are the only ones that bite you, that it only turns it on once a female has had the audacity to suck a blood meal out of you, okay? So otherwise, the genes are off. It's not ever on in a male, but if a female bites you, then it gets turned on, and there are many genes like that get turned on in a female mosquito, those, for example, that are needed to digest the blood. So he took a genetic element like that gives strong activity of a gene, any gene that's plugged next door, in response to the female having eaten a blood meal. So again, this thing will be will have almost no effect on the mosquito in the case of mosquitoes that aren't actively feeding, or in the case of males. But when a female does feed on blood, this again turns on these genes, which we call effector molecules, which are basically just antibodies, genetically encodable antibodies that bind to the parasite and prevent it from being transmitted. And in Tony's case, there are actually a pair of them that are represented by this black line. So it's kind of like a combination therapy that completely 100% blocks the transmission of malarial parasites in mosquitoes that carry this element. And at the end of the paper, where he had exhaustively demonstrated the effectiveness of this element, the very last line, he said, if this were coupled with a mechanism for gene spread, malaria resistance transgenes could become a self-sustaining disease control tool. And so when we saw this, we go, whoa, that's us. So maybe we could combine our widget with his genetic device. So we gave him a call, and he was very enthusiastic about doing that. And to make a long story short, when Valentino made a genetic construct that was then put into the mosquitoes by Tony, it transmitted through mosquitoes with to 99.5% of the progeny. This when it's transmitted through males, which is a point that I'll be happy to get to in the end if people have questions, but showed proof of principle of how well such a genetic element could transmit. And it was a very large piece of DNA, one of the largest pieces of DNA I think that have ever been propagated in such a way. And so this sort of gave proof of principle to this idea that you could bring in an immunizing gene cassette. And he, in fact, showed that those genes that are the immunizing genes could become activated when those females that carried it fed on blood. And so the idea is that you could start off by introducing a tiny number of mosquitoes, 1% of the total population, into an area, and then within 10 generations or about one season, in principle, they could spread throughout the entire region. And to put this in perspective, people who use sterile insect technology typically dump as much as 10 times or more numerically animals into an environment compared to those that are present. So that is something that people have been doing for almost 50 years now in the state of California to control medflies, for example, or other pests. So this is like 1/1000th the number of animals that would be added to the environment compared to those types of genetic insecticide treatments. So, of course, a concern with this is that since it can spread so pervasively, and one important element of the Tata Institute for Genetics and Society, which is the institute that has been supported by the generous gift we have. The S stands for society, and so an important element of that is trying to understand sort of the ethical issues and safety issues. So from a science point of view, one of the things that people are worried about is, well, how would you stop an element like that can keep spreading it ad infinitum? How could you potentially limit that? And so one idea we have is something we call an eraser. It's a backronym, so you really don't have to look at this at all, but it erases the gene drive element. And the key of it is that it contains guide RNAs that provide the information of where to cut, but it contains itself no enzyme, the Cas9. It contains no Cas9. So the way this works is that this gets made so that it's exactly in the same place of located in exactly the same part of the genome that the drive element is. And when one of these that mates to one of these, and you get a situation where one chromosome comes from a drive parent and one chromosome comes from an eraser parent, what happens is that the guide RNA that's made by the eraser combines with the Cas9 provided by the gene drive to cut over here. This other one also combines with Cas9 to cut now on this side, and the result is they cut out the gene drive element. And so if you imagine this piece of DNA just kind of floating away, which it would, then this makes a gap in the chromosome on the gene drive chromosome, and it gets repaired by copying through the eraser and then inserting this information into the hole there, as did the gene drive initially in the sort of naive chromosome. And the result then is that you get both copies of the chromosome carrying the eraser. And now there's no Cas9, so it kind of puts an end to that run. And so you can kind of model it in the following way. You could have a gene drive that sweeps through a population in one season or so. As it gets close to this point and you want to stop it, you then release an eraser. An eraser can drive through a gene drive population with the same kinetics as a gene drive can through a naive population, and at the same time it replaces it. So it copies and replaces. And then at the end, you're just left with the eraser, no gene drive. And the erasers can also be designed to carry the anti-malarial effectors, so that now you've replaced the gene, you've allowed the gene drive to spread throughout a whole area. Then you follow that with the eraser, and now at the end of the day, it carries the anti-malarial effectors, but it freezes the process in place. So one of the things where this is important from a social point of view and a legal point of view is the Cartagena Protocol is an international agreement that says that no genetically modified organism should cross an international boundary without the approval of both nations, which is, of course, a very sensible rule. And so this would allow potentially for a situation where you had that kind of problem where one country was okay with the idea and one country was not. Now, I'd just like to end by talking about what is the idea of our implementation plan. So the idea is to follow the recommendations of the World Health Organization that Tony James, over his long career, has played quite a role actually in shaping himself, which is that it's a phased implementation plan where initially work is done in the laboratory to show that the organisms have the design properties that are needed. And then in a go, no-go decision, one then can proceed on to the next phase, which is cage trials, field trials where the animals are kept indoors in cages, or in confined field trials, like, for example, on ecological islands, perhaps literally an island in the middle of the ocean. But in any case, areas where one can control the process and mitigate it with standard pesticides, for example, if a problem should arise. And then again, in a go, no-go decision based on the results of these confined field trials, proceed on to staged open field releases, and then eventually into implementation. And throughout this entire process, the idea is to have regulatory oversight, community engagement, and communications as an integral part of that whole process. And I'd just like to end by pointing out that the TIGS mandate involves, of course, an emphasis on vector-borne diseases, but that there are other ideas of applying this general copying process, which we refer to as active genetics, to accelerating agricultural research, reducing antibiotic resistance, potentially coming up with ways of treating cancer or aiding existing approaches by making them much more efficient. And then, of course, the ethical and public policy implications of active genetics as well as part of the package. And that this is something that has now become a very active area of engagement, of academic engagement at UCSD. And that within the science component of it, for example, not only are experiments being done in mosquitoes and fruit flies, but also in human cells, again, hoping to have an impact on diseases like cancer, mice and fish to create models of human disease in vertebrate animals, mammals in particular, and in plants to potentially increase the endow plants with better characteristics more rapidly for agriculture, and then experiments in yeast to understand the mechanisms by which this whole process works. And these people are actively involved in our active genetic program here at UCSD, and we also have people being recruited to the social component of it. So I think this is a very exciting moment at UCSD for this whole field. So I'd like to turn it over now to Elizabeth.
Okay, yeah, thank you. I'm going to talk a little bit more just about the general disease and specifically about malaria in South Asia. And I just decided to put this one up because this is actually one of the reasons I work on malaria. People often say, well, why don't you work on cancer and so forth? And there I am in 1968 sitting there with my sister, and this is about the time that I actually got malaria. My father was an anthropologist working in Malaysia at the time. And so I've retained a big interest in tropical diseases. So malaria is one of the world's deadliest infectious diseases, along with TB and HIV. And the thing that all of these diseases have in common, and which makes them particularly difficult to if you're thinking about eliminating them, is the fact that we have no effective vaccines for any of these. So unlike smallpox or something like that, you can't use a vaccine to get rid of these diseases. You have no real natural immunity. You can be exposed to malaria, and if you'll eventually develop tolerance against malaria that's in your area. But if you move away for a number of years and then you come back and you don't take appropriate prophylaxis, you can get malaria again, most likely because malaria parasite is a little bit like the flu virus, and that it's often different from season to season. The parasites are always changing. Not all of it, but they have chunks of genes that are involved in antigenic variation that are often shifting. You also have latency and recurrence, and this is particularly a problem in India where you have a lot of Plasmodium vivax malaria, and the parasite can go into your liver and it can hide there for long periods of time. And so you can go and treat everyone. You think you've solved the problem, and then someone has a relapse. They don't necessarily go and seek treatment for this relapse. They may not even know that they have malaria. They just have a fever. they get bitten by a mosquito during that time, and then they can spread the disease to other people. So this is a particular problem for elimination. And the other big problem with malaria is that you have a lot of drug resistance, we had a new class of medicine, artemisinin and combination therapies, that were introduced about 15 years ago, and they've been very effective at driving down the death rates. However, there's now pockets of pretty severe resistance, especially in Southeast Asia. I don't think it's been cataloged quite as much in India, but near the Thai-Cambodian border, there's quite a bit of artemisinin resistance. So, malaria is a pretty severe disease. I think you said there were more than 2 million cases. Actually, the number is 200 million cases annually. So it is a very big disease burden in many parts of the world, in particular sub-Saharan Africa, but it also is a big disease burden in India. It kills currently about 1,300 children daily, and as Ethan said, it's spread by the bite of Anopheline mosquitoes. What typically happens with malaria deaths are primarily in children under 5. Falciparum parasites will proliferate, and they will cytoadhere to the capillaries in the brain. And once that happens, you can have essentially this creates brain swelling, and the brain swelling will push on the respiratory center in the brain, and then essentially the children will just die from respiratory distress. They'll just stop breathing, and it happens quite suddenly. And in many cases, the reason that children are dying is because they may not have access. It may be difficult for them to get treatment in time. For example, someone might have to bring a baby in, and walk a number of hours to get to a place, and by the time they actually arrive, the child has died. Malaria. Socially used to be found, malaria used to be found throughout the world, including in the United States. It used to be found in California and in the southern parts of the United States. That's why the CDC is located in Atlanta, was because it was a big malaria swamp. Through concerted efforts in the 1960s, they drove malaria out of the United States and out of Europe. And it is now really just concentrating in South America, sub-Saharan Africa, as well as in South Asia. But the fact that we were able to eliminate malaria from the United States and from Europe has led people to think, we can really think about maybe eliminating malaria from the rest of the world. And that is the stated goal for the malaria research community is malaria elimination. Malaria in India, in particular, remains a big problem, and you have two types of parasites. You have high transmission regions in just particular parts of it, but malaria exists throughout India. There's some questions about how much malaria actually existed, because if you actually take the government estimate, this is really small. The government was giving numbers like 384 deaths annually from malaria, which is actually heavily disputed by the WHO, because the estimated death rates here is something between 24,000 and has a range from 1,500 to 47,000. So the basic problem with this is malaria deaths in India are only recorded if someone actually has verified parasites in their bloodstream, and in many cases people don't check. So a lot of people are coming in and they're dying of fever in India, and it's not necessarily being recorded as a malaria case, so the number of cases may actually be bigger than people think. So one of the ways that they primarily treat malaria in India is with the use of insecticide treated nets and indoor residual spraying. And if you look at the expenditures for malaria treatment in India, the vast majority of it is going to insecticides and spraying materials, with just a small portion of it for human resources. And this would be people who would be going around from village to village looking for malaria cases, getting people to sign up for treatment. So this has been one of the, I believe this year, India actually started saying that they were going to enter the pre-elimination phase. And they've been making a lot of progress. And in 2013, the number of cases, reported cases, had been halved. So there are about 2 million cases in 2000. Actually, you were just probably talking about India when you said there were 2 million cases. But it's dropped down to 882. And they attribute a lot of the gains to the expansion of community-based diagnostic testing and treatment. This means for every village out there, you have someone that's the malaria expert that will go out, track people down, determine whether they might have malaria using what are called rapid diagnostic tests. So these are things I'll show you in a minute where you have a pinprick of blood and you put it down and then you check to see if someone actually has parasites. And this can be used if you don't have a microscope, for example, or access to a lab. There's been a lot of encouraging news coming out of India over the last few years. However, there are challenges to elimination in South Asia. And some of these are the spread of parasites that cannot be detected by rapid diagnostic tests. These are the diagnostic tests that I was showing you here. And again, you put a little spot of blood here, and it looks a little bit like a pregnancy test, and it will tell you if you have parasites or not. The problem with that is that the gene that's detected by this is actually fairly mutable, and it looks like parasites that are resistant to diagnostic tests are spreading through the population, because you can imagine if you've accumulated a deletion in the gene that gives you a positive readout in the diagnostic test, and you've lost it, that means that you're less likely to get treated, and that gene is going to have a selective advantage within the population. Another challenge to potential elimination in Asia is migrants and political instability. There's been a big resurgence in malaria in Brazil and in Peru because of the situation in Venezuela. Malaria is now, there's been a huge increase in malaria cases in Venezuela, and this is spreading into neighboring countries. So countries like Peru and Colombia have seen big increases in the number of cases that they have. So the situation in Myanmar will make it potentially challenging to eliminate malaria in India. There's also the emergence of drug and insecticide resistance. I'm interested in drug resistance, but insecticide resistance is also a major problem. They use DDT. And of course, we're also talking about, the strategies for using transgenic mosquitoes. But this is the current situation. And so what I've been particularly interested in is this question of whether we can actually get to elimination, to zero parasites using the tools that are currently available. So two of the big classes of antimalarials both come from natural products. So one of them is Artemisia annua, and this is the Cinchona officinalis, and this gave rise to chloroquine or to quinine derivatives. So people have heard about gin and tonics, of course. People took gin and tonics because of the tonic that's in it, and tonic has antimalarial properties. That was happening. This plant comes from Peru, and this is Artemisia annua, and this is also a fever-reducing remedy that's been known for almost 2,000 years. This was in the Chinese literature, and there's wormwood directions for taking wormwood extracts to get rid of fever that go back 2,000 years. So the active ingredient in these two plants is quinine, and then it's artemisinin, and these have been various derivatives have been made of these, but they're all basically the same chemical formula. So this is quinine, and then this is the active aminoquinoline ring here. They've just been attached. So there's a lot of new generation drugs, but many of them are just based on, are just derivatives of these old drugs. Now the problem with this is that, and the problem for malaria elimination is that these two compounds tend to only act against this asexual blood stage of the parasite life cycle. So Ethan told you when the mosquito takes a blood meal, it injects these sporozoites into your bloodstream. These will migrate to your liver, and they will set up a home in your liver, and they may come out right away, or they may come out 6 months later. This is where the latent phase, and this is particularly true in India. So neither of these compounds tend to touch the parasites that are living in your liver. And after that, they enter the bloodstream, and this is the stage that's associated with all the symptoms of malaria. This is where you have the fevers, the chills, and all of the deaths. So you can't tell at all if you have parasites in your liver. They're completely asymptomatic. And so you're not going to take a natural product that eliminates them from the liver because it's not going to make you feel any better because you don't know that you're sick in the first place. If you take something when you have a fever, you know you've tasted this plant, you start feeling better, and then you're going to take more of that plant. The other thing is that parasites have a sexual stage, and this is where we go into transmission. When the parasites identify that they're being stressed out, they decide, oh, it's time to get out of here. And when they do that, they decide to differentiate into these male and female gametocytes. These are eukaryotic pathogens. They have a complete sexual cycle. So they decide they're going to differentiate into these gametocytes, and these gametocytes are the only form of the parasite that can actually survive in the midgut of a mosquito. They don't get digested. If the mosquito takes up some of these, they get digested by the mosquito, but if they take up gametocytes, the gametocytes are resistant to digestion. Soon after they realize that they're in the mosquito midgut, they differentiate. They turn into essentially sperm and egg in the mosquito midgut. They find each other, they mate, you have a brief meiosis, and then the parasite will migrate across the wall of the mosquito midgut and set up a cyst, and then it's transmitted the next time the mosquito takes a blood meal. So what we've been interested in here in my laboratory is trying to find next generation medicines that will not only eliminate these asexual blood stages, but will act to block transmission and to prevent you from getting malaria as well. And that's been a big part of our research over the last 10 years. So this idea of going and finding new drugs, well, you could take a very rational approach and try to find what's called a drug target, and then you could try to crystallize this drug target, and then you could do very elegant docking, it's just like trying to find the key that will fit in your lock. And it's a very beautiful process, but it requires you to know a huge amount about, how you want to kill the parasite. You need to know which genes are essential. And we decided that we would just go and back to the sort of very simple older approach, which is a little bit like Paul Ehrlich's magic bullet approach, where you just take lots and lots, hundreds and thousands of chemical compounds, and you test each one of them, and you find something that kills parasites. And they used to do this back in the 1910s and 1920s. They would make a batch of a compound, and they'd inject it into a canary, and then if it cured the canary of malaria, then they knew it was good. We can't really do that, and the problem with that is that it's extremely low throughput. Maybe you can go through 100 or 200 compounds. So we have invested in my laboratory trying to automate these processes so that we can go through millions of compounds with the idea that we can more readily find the magic bullet that's going to be used to cure malaria, or at least prevent malaria. And so we've developed 3 different assays here, and this is a 1536 well plate. And what you would do is you would spot out a little tiny nanoliter. You can barely even see this with a dissecting scope onto this plate, and each one of these would be about the plate would be about that big, and you'd have 1,500 wells in it. You'd spot out a very tiny amount, and then you would either put in blood stage parasites, which is shown right here. And up here we have liver stage parasites that are growing in hepatocytes, and over here we have dispensed out gametocytes. And then we put our compounds in, and all of this has to be done, using automation and engineering, and then we wait a couple days, and then we detect to see if we've killed the parasite or not. And here we're detecting whether we killed it by looking at an increase in DNA content. Here we have a luciferase assay, and here we're looking at the ATP that's produced by the parasite. And so we can get readouts now for hundreds of thousands of molecules. And again, I mentioned that this is a lot of engineering. These are a lot of the tools that we've set up at the Skaggs School, working with Jim McCarrow. And we have liquid cell dispensers and automated liquid handling, machines that can drop out tiny, tiny amounts of these compounds, which are actually quite precious. Recently, we've been starting to look for compounds that we can use to create what we call a chemical vaccine. So what our vision here is that we've created a compound that will protect you from getting malaria. And the reason we want to protect you from getting malaria is because in the initial stages of the infection, you only have 100 parasites present. Well, if you're trying to cure the disease after you've got it, you have 1 billion parasites present. So the potential for drug resistance is much, much higher when you have 1 billion parasites present. And so we've been going through hundreds of compounds. We've screened about 500,000, and we've gone down to a series of compounds that we now think are starting points for the next generation of malaria drugs. This was all work that was done with UCSD, and a lot of it was undergraduates here dissecting mosquitoes. That was a big part of the challenge. Another cool thing about working on malaria is that the compounds that come out of this, since they were donated ultimately by the Medicines for Malaria Venture, this is what we call open source drug discovery. And our objective here is to publish the identity of all of these compounds and to put detailed information out about them so that other people who have a chemistry background can just then order them up or make them and then initiate their own course of drug development. So these are screening hits here. They're not perfect. They're not suitable quite for putting in people, but some of them are very potent. But if someone has a bit of a chemistry background, they will hopefully be able to look at this paper, identify this molecule. It has 100 nanomolar activity. We'll have detailed information about it. And then they can take this molecule and then try to create some modifications on it that will make it suitable to make it more drug-like. And so is this working? Well, we're working on finding a new prophylactic compound. However, I started a lot of this work when I had a joint appointment in, I'll skip that. These are some of the compounds that have come out. When I had an appointment at the Genomics Institute of the Novartis Research Foundation, and we did our first asexual blood stage screen there. And using this same general approach, we were able to create the next generation anti-malarial drug. This is a compound that has a completely new mechanism of action. It's different from anything that was published previously. And it has, with a few rounds of medicinal chemistry, it was developed into something that's actually orally bioavailable and has the potential to give a single-dose cure. So that's a huge advantage over the existing medicines. And this has it's no longer in my hands anymore, but it's progressing into clinical trials. And the nice thing about K609 is that it's able to cure patients in a fraction of the time of all of the other drugs that are currently given. So it's about, it will eliminate all the parasites from your body in 12 hours. So this is being further developed, and it wasn't just one compound. We have a second compound that also came from our initial screens that also is able to eliminate malaria parasites in clinical trials. So that's what we're doing. I don't really have time to talk about our resistance selection. A lot of this work on our drug screening is happening here at UCSD. We have a team of undergraduates. And in addition to doing our tests, and we do a lot of biology as well, and we also test compounds for everyone all over the world. So things get sent to us, and then we tell you whether it has potential anti-malarial activity.
Perfect. Thanks so much to Kamala, Vineet, and others for organizing the event and inviting us. I feel pretty unqualified to talk about malaria relative to the kind of science expertise we've seen. But what I thought I would do is just provide a little bit of a policy perspective, which is what I do. There's some duplication with what Liz presented, so I'll go through that faster. But what I thought I would do is just, talk about just the basics of measurement and economic costs quickly, and then talk more about where my work on public service delivery has some insights in thinking about the optimal approach, because a lot of my work is focused on measuring the quality of public service delivery in education and health, including a lot of primary work just on the quality of frontline delivery. So, and then the goal is to then use that as a lens to think about as we think about the policy choices facing the country between kind of deploying the traditional approaches versus thinking about something completely out of the box. I think my main additional lens is that what might be optimal, say, in Florida, where a Zika kind of referendum on active genetics failed, might be very different in a place like India, where any conventional system has to be mediated through essentially a dysfunctional public health system. So I think that's kind of the main goal of what I'm trying to do here, and then we can talk about other issues in discussion. So there's some overlap with what Liz talked about. So the WHO numbers are in the range of 200 million with about 600,000 deaths. In India, about 95% of the population does reside in endemic areas, and the incidence is disproportionately in more disadvantaged areas, in tribal, hilly, more inaccessible areas, low-income rural areas of eastern and northeastern states. So if you just look at the distribution of the annual parasite incidence, it has come down a fair bit, in large measure due to the success of the conventional strategies. So it's not like these have not been working, but there's still a very substantial magnitude of the problem, and that belt you see there is kind of the predominantly tribal hilly belt that also has is also subject to the worst kind of Naxalite violence and domestic violence and internal conflict, and so that makes it harder for the formal machinery of the state to reach in some of the most vulnerable areas. So you kind of see that incidence almost tracks nicely, or not so nicely, but tracks kind of accurately where those challenges are. So as Liz alluded, so these are the death numbers in terms of fatality. This is also coming from WHO in India, about 20,000. But as Elizabeth was talking about, there's actually very, very substantial measurement challenges. And this is, again, politically very fraught. And this is a big problem for anybody working on healthcare in India, which is that the entire the policy architecture runs through a lens of a public system and kind of almost ignores, and I'll talk to you a little bit on the curative side as well. But essentially, the government will only record this in the statistics if they're kind of in the official, confirmed by a test at a government health facility, while the vast majority of patients will either see a local private doctor or never see a health worker, or if there's a fatality, you really have no idea. And so they just simply don't appear in the government's radar, and so most serious observers kind of just simply discount the government data. So the question then is, what do you do? It's still an incredibly hard measurement problem. So one kind of very powerful recent initiative, though it might be biased the other way, is because there's no kind of tradition of autopsies or pathology in India. So you've got millions of deaths, and there's very little understanding of what caused these deaths, though in many cases they are multi-causal. So there has been this very ambitious effort called the Million Death Survey that's just literally going and doing ethnographies, and I think the New York Times had an extensive article on this last month, and just do verbal autopsies by talking to family members and trying to recount all the symptoms and piecing together all the information in a piece of kind of literally ethnographic forensic detective work to come back and classify what they think the cause of the death was, right? So if you kind of use that source, which is obviously not biased by any kind of reporting biases of coming into a public clinic or being tested, these numbers look much higher, and in the range of over 100,000. Now, of course, this is likely to be biased upwards. Because the problem, and I'll come back to, the problem of overtreatment, right? Which is when people get a fever, they will often think that they have malaria. And we've seen this, particularly, there's a lot of good work on this in Africa, some in India too, of, people just going in and asking for antimalarials when they have a fever, and doctors often just giving it to them because that's kind of what the patient wants. So we've got problems on both kind of underreporting through the public system, but overreporting if you just rely on self-reported morbidity. But this is a global problem, right? It's a fundamental measurement problem that exists whether you're in India or Africa. And the WHO's estimates are only about 14% of cases get detected and reported through the official surveillance system. So I think So that's in terms of magnitude. Now, if you go to economic costs, there's three main components of the costs. There's the treatment costs, obviously, like of going, seeking care, seeking the doctor's fees, the medicines. But it turns out that the much bigger cost is the economic cost of not working. And so that's where the bulk of the cost is. And then there's, of course, the additional cost of premature death. So the best recent estimates of this, there's a paper by Gupta and Chowdhury, the Institute of Economic Growth. And so what they do is they use the National Sample Survey and use household data to then kind of measure at different quintiles of the income distribution how much households report spending on each of these segments. So the malaria is self-reported, right? So they have to rely on that because there's no other data set that matches source of morbidity with any meaningful economic numbers to estimate these costs. So what they're doing is they're taking the self-reported cases of malaria incidents and then looking at the NSS data to quantify the economic cost, and then they're multiplying it by the WHO incidence estimates because they trust that, they think that's more credible, and come up with an estimate of the economic cost of about $2 billion a year. And this is a nominal term, so in purchasing power parity, which is what most serious development economists will use because of variation in the cost of services, if you do a PPP calculation, that number is going to be bigger. It's going to be more in the range of $6 billion a year in terms of what is the value to India in terms of rupees. So I've already said the majority of the costs are driven by the opportunity costs of not being able to work. And this is almost certainly an underestimate because it will go up with economic growth because the opportunity cost of time is what's driving this. Okay? So that's just economic costs. So lots of other studies and other outcomes because, this is just essentially, these are short-term costs, right? So again, this is going to be an underestimate because it's not looking at the kind of patterns Liz was talking about, the increased risk of long-term mortality, increased risk of, so there's gazillions of studies well identified, not just correlational epidemiological studies. So I think a lot of the work that the economists have done has been trying to identify what would be a quasi-random shock so that you're not just confounding a bunch of unobserved variables. That there are randomized studies, evaluations documenting effects of malaria on cognitive ability and school absenteeism. So that's effects on kids. There's effects on long-run health outcomes, labor force participation, lower wealth, lower educational attainment. And then some of the nicer kind of long-term studies, what they use is they use the fact that even if you have a nationwide rollout, say, of an anti-malarial campaign, that's what the Cutler et al. Paper does, you can then look at differential impact as a function of pre-existing morbidity. So if you have a nationwide campaign, the effects should be bigger in areas where the disease prevalence was bigger in the first place. So that gives you a source of variation because the nationwide policy could be correlated with other health initiatives, right? So there's a lot of naive estimates that will say, hey, we rolled out this nationwide health program and then, we got these effects. But that could just be driven by economic growth, driven by lots of other unobservables. So you start doing a little better by looking at these kind of differential impact studies. And then, of course, there are all the long-term correlational studies. Okay, now, what's interesting here is that, malaria eradication is a stated priority for the government. So it's not like, they need us to come tell them this is important. They recognize it's important, right? And they are trying their best. And if you look at the national policy, right, it does say that there's a vision of eliminating malaria nationally and, all the good things, right? The problem is, and this is what I want to spend a little bit more time, is that, India, like for people who haven't really experienced the Indian state on the ground, it's really nothing like you've ever imagined, right? What is written on paper, what's on policy might have nothing to do, right? With what you experience on the ground. And I'll just give you some stats about how, you can have the most beautiful, grandiose policy goals at the national level, but to the extent that it is implemented and mediated through a pretty much dysfunctional public healthcare system, kind of completely changes what is optimal, right? So what is optimal in the US is and this is, I think, a problem for a lot of technical folks because, and I see this even in the context of, say, agriculture research, right? So there might be something on a test plot that you will say is an optimal combination of inputs. But then if you go take it to the field where you've got completely different set of constraints, what is technically optimal might be very, very different from what's actually implementable. And the same principles apply to kind of healthcare, right? So we know the difference between efficacy and effectiveness trials, but I don't think we've really taken the same level of care in saying, here's our solution. If you push it through this broken system, what happens? So let me now spend more time on these facts because that's where most of my research has been. Okay, so just a little bit of facts on the public health system. The default policy approach, which follows WHO guidelines, right, is to have public clinics that provide free or highly subsidized care for those who seek it. So in theory, on paper, we have universal free healthcare, right? Because there's a system of primary healthcare centers, community healthcare centers, district hospitals, where in theory, these are supposed to be staffed with qualified staff and any patient should be able to walk in and get care. I mean, that's kind of what the system looks like on the books, okay? Now, the policy, and they're supposed to be staffed with qualified doctors. Now, the policy discussions, there's a large emphasis on strengthening this system all the way back from the Bore Commission report like in 1946 to the recent National Rural Health Mission. The focus has been on pretty much kind of pumping more resources down this public system, right? So build more facilities, hire more clinics, hire more doctors, and provide more medicines, that's kind of the default focus of the public system. Till you go look at the household survey data, right? And the picture is exactly flipped on its head, which is that about over 70% of the first contacts of primary care in rural India are with the fee-charging private sector. Okay, so even though you have free public health care, most people are walking into clinics and paying like no insurance, right? 100% out of pocket, for healthcare. Now you might think, okay, maybe these private guys are better qualified. No, okay? 80% of them have no medical degree, right? So these are complete quacks, okay? So what do you call BAMs, BIMs, bums? So you've got these alternate medicines and 77% have no qualification. 18% have some alternative qualification, which is Ayurveda, integrated Unani, homeopathy. India, we believe in a multitude of gods. We believe in a multitude of healthcare systems, right? So there is a ministry of alternative healthcare systems and they have equal status, okay? So you have all of these guys, but the point is that only 4% of these private providers actually have formal qualification. So from an economist perspective, this is an incredible puzzle, right? Because in theory, the public providers are more qualified. They offer free services, but have under 30% market share. So what does it say about the quality of your product that you can't even give it away for free? Okay. Now, if you then look at the discourse on why this is, obviously the critiques from the left and the right, everybody for the most part agrees on the facts, but the critique from the left is mainly that you're not spending enough on the public healthcare system. Okay. So the critique from the left will be look at Tamil Nadu and Kerala. These are states that have spent much more in their public healthcare systems and that they do much better. The critiques, I think, more from the right would be would be that the bigger problem is that you have a completely unaccountable public sector and there is really very little return to pushing money down, that kind of unaccountable system. Now, being agnostic to left and right and just going and looking at the data, right? A non-trivial amount of my work kind of has focused on documenting these accountability challenges, which is what I'll talk about. And in one slide summarizing a bunch of studies. So one of the earliest studies I'd done in this area was back in 2003, where we did a nationwide surprise visit of kind of public health clinics and primary schools in 20 states. So we covered about 3,000 schools and over 1,500 of these primary health facilities. So we just made surprise visits to these places, had rosters of the staff, and just went and counted, who was there. So it's as simple as that. And we found that about 40% of doctors were absent on any given day, okay? So absent. This is nowhere to be found, okay? And then, so that's fact number one. Fact number two, we then went back and, we did a very careful audit study where what we did, and I'll tell you a bit more about that, right? So the problem with measuring healthcare quality is that the traditional approach is you measure facilities, okay? But we know the facilities are not correlated much with outcomes. Then you go measure staff, you count people, but that's not very useful if they're absent, okay? So what you need to do is get an actual measure of quality of care, okay? Now, people have tried to do the kind of medical vignettes of the sort you'll get in your board certification. So you'll go and give the doctor a set of vignettes, a set of symptoms, and saying what's going on, right? You evaluate on the pathway of questions. But we have a lot of evidence that there's a huge gap between knowledge and practice, okay? So what they know is not what they do. So what we ended up doing was kind of doing this kind of Herculean 6-year project with undercover patients, doing an audit study, right? Where they were trained in standardized patient protocols of the sort that you have in medical schools in the US, except that they were trained with, in fact, one of India's leading medical anthropologists to help them kind of convey the diseases in the context of the local language. And this was done across 5 districts in Madhya Pradesh. And so we did this undercover audit study, and the results are kind of published in a bunch of papers, but the headlines are, for example, that 70% of public doctors have a completely illegal private practice. And then over a year, what we managed to do is we managed to send the same case to the same doctor in both the public and the private practice, okay? So using different people, of course, but it's the same case. And what you can document is how systematically higher the effort and quality of care is when you see that doctor in his or her private practice, okay? Now, But the problem is not just that. The problem is that the typical doctor, and this is true in both public and private, spends less than 2 minutes per patient per consultation. Now, this is true in not just the audit studies, but even the observational studies. And this is not because the system is so overburdened that they have a queue of like 100 patients that they need to kind of see people every 2 minutes and push them off. Most of the time, they're just sitting and twiddling their thumbs. Like, I mean, it's like you come, here's 2 minutes. Like the average encounter has one question, one exam, and you'll usually get a bunch of medicines and be sent out. Okay, so this is what happens in your typical interaction with a healthcare provider in rural India. So the rate of correct diagnosis in our setting was under 4%, okay? And now, there is kind of underreporting bias here because for the most part, the doctors don't feel the need to pronounce a diagnosis, right? You have this audit study. So we tried this little experiment randomly in half our sample at the end of the encounter when our standardized patient turned around and said, oh, doctor, what's the matter with me? Most of the time they get yelled at and say, you don't need to know, okay? So even conditional with all those biases, you're getting about 10%. And then what's so interesting and what's so vexing about healthcare, like most of the advances in economics, a bunch of Nobel Prizes in theory for asymmetric information have come from healthcare, right? Because markets don't work so easily because of asymmetry of information and because it's a credence good that you rely on the doctor to tell you what you need. And so the market kind of breaks down, okay? But and consistent with that, what you see is that there's substantial overtreatment, right? Over 80% of our cases in our audit study received unnecessary medication. But what's even more interesting is you see that even in the public clinics, okay? So why does the public guy give you excess medicine when he's not making money out of it? It's because mostly what the patient is looking for is they're looking for symptomatic relief to be able to go back to work, right? So this is a setting where there's no unemployment insurance. A day you don't work is a day your family doesn't eat. So even the public system, it's not that they're completely heartless, right? But the returns to accurate diagnosis and the returns to then giving you the right medicine are much lower relative to the cost of effort, relative to just giving you a bunch of relatively strong painkillers and steroids and making you feel better and go back to work. So that's kind of what the modal patient-doctor interaction looks like in both the public and the private sector. So that's kind of the context in which we are. So something Ethan didn't talk about but in the slide was we talked about antibiotic resistance, right? But that's where it comes from. I mean, it comes from the fact that, the average encounter has 80% of cases have excess medication, and that applies to antibiotics as well. So now, the health system is not completely dysfunctional, right? It does work better when it's on a mission mode. So if the entire system top-down focuses on one or two metrics and tries to make that happen, then you can kind of marshal the entire machinery of the Indian state to deliver those things, right? And so India works well in mission mode. So we kind of run the most amazing elections in the world, right? The Russians haven't quite hacked it yet. Okay, maybe just because they're offline, okay? But these elections are an incredible feat, okay? And so there are things that the Indian state does amazingly well, but the day-to-day functions of governance that are not monitored with that kind of intensity just simply don't function, okay? Now, even when you have the intensity of mission mode, look at something like vaccine immunization. So I think polio was this nationwide pulse polio campaign that kind of succeeded, but if you look at kind of success in immunization, which is kind of much more basic and easy to deliver relative to something like diagnostic and curative care, even on immunization, if you look at the coverage of total immunization, the children who received all of their vaccinations that they have to, there's still like substantial gaps, particularly in the poor states of UP, Bihar, and Jharkhand, right? And so, given just these challenges of the capacity of the Indian state, I think the implication for us here when we think about what does this mean for us as we think about the optimal approach to go and address something like malaria. So I don't know the sciences. All I can tell you is whatever approach you have has to be mediated through this dysfunctional system. And so if we're going to make progress, we have to account for that. So when you think about, active genetics clearly has enormous potential, but also generates a lot of public apprehension, right? But my own assessment of this is that if there's any place in the world where you would kind of put your thumb on the scale towards an active genetics-based approach, it is India, right? So, a Zika referendum, the active genetics approach could fail in Florida, but the curative options in the US are still pretty good, okay? So you might not be able to eradicate Zika, but if you catch a case, you will still be taken care of pretty much at, say, the 80th percentile of what is humanly feasible, okay? In India, you'll be lucky if you're at 2%. Okay, so given that gap between where the global knowledge frontier is and what we can provide to the average Indian citizen, the ethical and moral case that can mean for thinking about solutions that essentially bypass the dysfunctional kind of public system become much more compelling. Okay, so now this is not to say therefore that we must do this, but when we eventually come back and weigh these options, right, like I think this pushes us, me at least, as kind of a public policy person, to feel a little bit more optimistic about, or not just optimistic, I think when I think about the way forward, and we'll talk about this in the panel, the first time I heard about active genetics, you feel this kind of tingling feeling in your spine, right? This feels like one of these Oppenheimer moments, right? I mean, that we've unleashed something that can't be put back in the box. So I think I see this as two sets of issues. The first is, as a community of experts, who is concerned about the global good, do we have a view on what's the right thing to do? And then conditional on having that view, how do you then think about what is the right level of public outreach and acceptance to then get something that passes standards of democratic ethical validation as well as international and global consequences. So let me stop there.
So I'll now invite all the panelists to come and take center stage, and we will have we'll invite the panelists themselves to ask a few questions and offer some commentary at first, and then we'll open up the floor to questions from the audience. But please. So perhaps we can go in the same order as we had started, and maybe Ethan, if you want to offer any kind of commentary, and then we'll go to Elizabeth and Karthik.
Well, I found a lot of the points that Karthik made very interesting, and one of the things that's obvious is one has to think about the problem of what is the ethical way of delivering a technology that's somewhat difficult to explain to people who have an education that limits their ability to understand the technical nuances of how something works. And we've talked about it only at a very superficial level among ourselves, but one of the things that just take as a kind of example, just as to put it in a ballpark of thinking about it, is just what in principle is the ethical sort of approach to bringing the information about a vaccination program to people who need to have it explained in terms of what exactly does the vaccine do, what are the risks of the vaccine, because of course, some people who get vaccinated actually die, right? I mean, that does happen. So you have to explain that fairly, but on the other hand, you can't really get into the details of how a vaccine works and why exactly the immune system in some cases might have an adverse reaction and so forth. So in the same way, given the nature of the system as it works in any place, India in particular for us, what are the paths to connect with people fairly, to give them, on the one hand, the information that they need to make a decision, like is it worth it to take the risk that we might not know exactly what a gene drive would do to the mosquitoes, so the mosquitoes might not be quite the way they were, and the nature might not be quite the way it was with regard to the animals that need mosquitoes, but on the other hand, we hope that we would be able to reduce the incidence of malaria. But is that fair? I mean, those are the kinds of questions. And then also, how do you work through the system, which I have to say, that's a sobering little description there, Karthik.
Yeah, I think, in general, development economists over the past 10, 15 years have started, we used to spend a lot of time on issues of, so taking a step back, right, when you think about why we expect poor countries to grow faster, there's basic economic theory tells you there's two reasons. One is you have less capital per worker, and so you have higher returns to capital. But second and more important is you're behind the knowledge frontier, and so it's easier to kind of copy what's successful than it is to just innovate from scratch. So Angus Deaton, who won the Nobel Prize in 2015, has documented through what are called the Preston curves that life expectancy in today's developing countries are substantially higher than what they were when today's OECD countries were at the same level of per capita income. Right? And that's true for every social sector outcome. So you take it for education, you take it for health. So the good news is that when there is discovery, there is diffusion, which kind of facilitates that catch-up. Now, The bad news is there's also a lot of $100 bills lying on the sidewalk. And so a lot of the work that economists have been doing is studying technology adoption at the level not just of kind of agriculture or manufacturing, but at the individual adoption of kind of, say, insecticide-treated bed nets. So, there's been a lot of work on this, and along the lines of what you're saying, that reducing the transaction cost, reducing just the number of times you need to do something, so I guess my point is that the work on those dimensions will continue to be useful, regardless of what happens in gene drives, because it's going to apply for everything from contraception to taking other medicines. So I don't think that these are either or approaches. So in that sense, the conventional view, gene drive is really like kind of a deuce machina, right? Like we came out 2 years ago to say like, wow, suddenly like we'll solve all these problems. So I think practically, turning back to the political and practical issues. I honestly don't have a read right now about how this will play out. I think as far as the TIGS is concerned, we assure ourselves by the fact that our counterparts are kind of the senior-most people in India, and we trust them to kind of and follow their judgment. So one of the things I think Suresh and I are planning is to go meet with the top kind of people in the life sciences group in India and saying, assume the science is ready in 3 years or 5 years. What preparatory work would you want done on kind of the society side, ethics side? What kind of plan do you want? And I think our plan is to let them drive it and then support it to the extent we can.
Right. One thing that, I recently read a book on eradication program for smallpox that took place in the 60s and 70s. This guy, William Fogg, who was one of the workers from the CDC who contributed to that. And he described the final challenge was India. And there was an amazing battle that remarkably was solved in just about a year and a half, where it went from, massive cases of smallpox to eradicated because they had the right implementation. But the right implementation involved a close collaboration between the CDC and the WHO from outside. And then, of course, a huge and expansive array of people within India that were sanctioned by the national government and the local governments, but then were staffed by 100 and 200,000 workers that would go out and identify cases. And you're talking about how there's a similar kind of infrastructure that is perhaps being built, and how maybe this health system is at its best when it's given a mission. So that was kind of also something that resonated a little, is that maybe that would be another thing to try to recapitulate the magic of that time when it became clear that there was a possibility of eradicating smallpox. And one of the things that Fogg pointed out in his book, it's a wonderful book called House on Fire. Anybody who's interested in reading it, it's an incredible story. But one of the things that he pointed out that was a very important component of it was just the idea that people believed it could be done. And because they believed it could be done, people who normally would have more dysfunctional relationships with one another opted out of that mode and instead, demurred and cooperated. And I think that's an interesting kind of psychological phenomenon that could happen at that sort of super level that depends on this kind of, idea that there's a common mission, and then recruiting enough people into really believing it in some way. So, I don't know how one does that, of course.
Thank you. So we'll open the floor to questions from the audience. I only request that if you ask a question, just please identify yourself, because we are a diverse group and not everybody knows each other. So are there any questions? Okay, maybe I can, yes, Ramesh, please.
So on an optimistic note, I don't see why the common person in India will experience any negative consequences of gene drive-driven malaria eradication programs. They might be disappointed that you're not going to eradicate mosquitoes because they get bitten by them. But it's sort of invisible, unlike many other things where you actually feel the adverse consequences. So I feel optimistic that you can do this without carrying the whole nation with you. But on the other hand, genetically modified vegetables have been hotly debated. And so I was going to ask you if there are any relevant lessons you might draw from how BT cotton and BT brinjal played out in India.
Yeah, so but that's exactly what I was going to say, right? I think And so I was talking, as part of the background for this, I kind of have been talking to the person who was driving gene drive policy in the Obama White House, and he kind of was at the middle of all the Zyka referendum and all that story, and he said, listen, you might think that the science is so obvious, right, but you're dealing with such emotional issues, right, that it's very easy to whip up a kind of frenzy of concern and just stop things, right? So there is certainly, I think, so 2 points, right? So one thing is that the fact that GM eggplant and mustard and all of these things are opposed, I think has two elements to it, right? There is kind of the concern about the genetic modification, but there's also the economic intellectual property side of that. So, the fact that the higher yielding varieties are typically patented by the big seed companies, and that has, that does create a bunch of conflicting forces. And so you kind of have this alliance of both the economic left and kind of the, just people who are worried about these Jurassic Park, for lack of a better word, right? Things that just get out of control. So I think in this case you're right. That I don't think there is a reason for popular concern. I think the challenge for us, and this is what Ethan was alluding to, is what constitutes the right norm of ethically informed consent in a setting where you're letting out something that's going to affect the whole country, right? So do you need a referendum? Do you say this is a democracy? So if a democratically elected leadership accepts, and if there's a certain amount of unanimity in the government, that it goes ahead? So again, those are questions which the Indian side will have to kind of support and weigh in on. But, I think those are the hard questions, right? And I think our plan right now, and I think Suresh will lead this, is just to be very transparent and right about what this is, right? But at the same time, be sensitive, but also be very realistic about why this is a big deal, right? That this is because you could leapfrog, right? So here's an analogy. So coming back to conventional versus leapfrogging, right? So, I grew up in an India where you still had to wait for 2 years for a telephone connection, right? So, and because the last mile challenge of getting universal telephony was getting cables out to every household. And you jumped that whole thing with the wireless revolution. So you kind of leapfrogged and caught up in 10 years to a level of coverage that was just inconceivable doing business as usual, right? So in a way, building out your traditional public healthcare system is a bit like that last mile, because you need every doctor in every village to be able to see every patient. And what you're doing here is offering a potential of a leapfrog, that circumvents those constraints. So I think the economic and substantive case is very, very strong. I think the main questions I think that Ethan and team will have to work on is kind of thinking about what are the escape valves if something goes wrong, right? And that's what I'd like to turn to you and ask you about. If you, given everything if you were to be like your own biggest skeptic, what are the pieces you would worry about the most, and how would you mitigate?
Well, I think, the thing that Tony and I have argued is the most likely scenario that we will have to deal with is that at the moment, Tony's anti-malarial cassettes can prevent the transmission of the parasite 100% at the level that the parasite is typically produced in nature. And but there are parasites that are variant parasites that can produce about 10 times more of the parasites in the mosquito, put 10 times more of a load on the mosquito. Now, why that isn't the case normally in nature, one can speculate on. As I understand it, malarial parasites don't really seriously impact the fitness of mosquitoes at the level that they're present normally. But if there were 10 times as many of them, I suppose it's possible that they're putting a metabolic drain on the mosquito, and that's why they're not at that higher level. Nonetheless, if you put a strong selective pressure on the parasite because you've got antimalarial antibodies that are binding them, and only if you have 10 times as many are you going to get away from it, and Tony has tested 10 times as many, and then a few of them get through, and the mosquitoes have to have like 3 blood meals to be able to, combat that. Now, of course, we're not far off from being able to match that 10x, but if you don't, then the worry is that you'd be selecting for a strain of parasite that would just be super numerous and then potentially super virulent as a consequence of that. Now, probably as in the case of when you apply pesticides and you get pesticide-resistant mosquitoes, that comes at a cost, that pesticide resistance, because if you take away the pesticide, the pesticide resistance disappears. So, but on the other hand, the reason you have it is because although it comes at a cost to the mosquito, it's even a greater cost to die from the pesticide. And this could be the kind of similar situation we face here. And so, the simple solution to that is to try to engineer the mosquito to begin with to be 10 times better than it is, so that we kind of put that roof so high that it doesn't easily evolve to that point in the period through which the relevant transmission or dispersal or dissemination of this cassette takes place, so that it's all over before the parasite has had a chance in that limited time to achieve such a large increment in its virulence or its capacity. But that could happen, and so it's important to have a whole bunch of other effector molecules waiting, and so we're trying to develop those as well. So there's things like that. I mean, nature always finds a way to take the Jurassic Park, analogy, and so I think you have to be attentive to those and see those. Now, of course, they're not problems that are unapproachable, though, because if they happen, then there are logical, rational things you could do. But I think that's the kind of thing you have to be careful about. It has to be thought about very, and discussed by people who are, looking at every angle of it.
So, I mean, the question I always have, when we talk about gene-drive systems is how well are we able to predict how many different breeding groups and how many different subspecies exist, and how well studied is this? Because you could in the laboratory make it so that it works great for Anopheles stevens eye, and then you release it out there, and then you find out in one particular region that there's 50 different little subgroups where they're all interbreeding with one another, and they don't like the person, the mosquito from this other group, and then you end up having to engineer different systems for 50 different subspecies, which obviously then makes it much more complicated. And so, are you worried about that?
Yeah, I am worried about that. And I think contrary to what some other people have worried about, is that you're going to release this and it's just going to go all over the world, and every mosquito everywhere is going to be transformed. Not that that was necessarily a terrible thing, but I think it's exactly the opposite. It's very much more like what Elizabeth is worried about, that the species structure, substructure will limit its spread from one to another. Now, there are things that can mitigate that. I mean, one of the important components of TIGS is to do a very significant sort of bioinformatic analysis of the population structure and test from different areas how well it works in different isolates to see indeed how well it will spread. But of course, as I've been recently informed from people who are ecologists, what you do in cages, even field cages, doesn't necessarily recapitulate what happens in nature. So one thing that they stress that's imperative is as quickly as possible to get it to some kind of very open, confined, but open release kind of paradigm, like an island or something, so you can really test that kind of thing. Because you're right, I think that's something that really needs to be looked at. And if you look at island ecologies, I mean, I don't know how much work has been done in India, in Indian islands, but Greg Glanzaro, who is working with Tony, has done quite a bit on African islands. You can have a relatively small island that has a lot of population structure on it. So those would be the perfect examples of places to test this, to see how much of a problem that is, and then how much you might have to do. But you're right. I mean, the worst case scenario, you'd have to re-engineer, which of course would be exactly the same construct, but you'd have to re-engineer it into every different subspecies or isolated mating group. If they made it all, presumably at some point you'd get it to spread from one to the other, but it could be slow. And again, I think that would be a very serious problem for people who are looking at the suppression approaches, because that would just stall it. But if you look at the immunization, it's more tolerant to that sort of thing, because you can just keep it in one population and just wait till it saltatorily jumps into the next one, and then it'll spread through the next one, presumably. But you're right. I mean, one has to do those studies, for sure.
Vish and then Siavash.
So one thing Karthik mentioned was the pushback active genetics card in Florida, and if I remember correctly, there were two counties, right? One was a bigger county in Florida, Monroe County, which actually supported, but there was another one, Key Haven, I think, actually opposed it, if I'm not mistaken. Because one of the things you're going to encounter is when you go in, even in the U.S., it's not accepted, and you're trying to bring it on us, kind of pushback, right?
And so we need to understand the nature of the opposition and how to surmount that, because that's going to be an issue. And so do you know, like, what, in fact, the much bigger county, Monroe was much bigger, if I understood, but Key Haven was a very small area that, that went against Zika, active genetics on Zika. And I didn't follow what happened afterward. The county of supervisors are supposed to make a decision. From what you're saying, maybe they decided not to go forward, although a bigger county supported active genetics. So so that's, I don't know if you know why they went to ban it and how to understand that. And also part B on this is, in terms of approach, one thing that's happening is there is a big push to use information technology, with the Aadhaar card and all of that. Within India, people, actually there was a survey done on where are people most trusting of their government, right? And you may have seen the results. India is one of the highest, 85% of the people trust the government. There is a new mood of optimism in India, despite what the scholars might say. So how do you tap into that, where people suddenly feel, just like what people in China felt 30 years ago? How do you tap into that? So
That's a big question.
One thing I'd just like to clarify with the Zika experiments, and actually Cinnamon Blossom on campus has done a study on that and analyzed it. And she wrote a paper that just recently got published. And if you want, I'll be happy to send that to you. But just to clarify, what they did in Florida was actually not active genetics. It was a standard insect sterile set of experiments. So the standard, that's been going on for 50 years in California is you take a very large number of males and you sterilize them by irradiation with X-rays, and then you release them, and they're sterile, but they can mate, and so they mate with the females, and then the females have no offspring. So Oxitec, this British company, actually they got the mosquitoes originally from Tony, which is an interesting little aside, but they had these mosquitoes that are genetically the equivalent in that they just simply are sterile, but they don't pass the trait down in an active way, which would be an active genetics thing. So it's just standard Mendelian inheritance, but if you raise them in a certain condition where there's a drug present, they're fertile, but then if you release them without the drug, they're sterile. And so it's just basically like treating with X-rays, really. But they have a transgenic element in them, so it's a GMO type of issue. And so the thing about what Oxitec did, which was, I think, a big mistake, they made a lot of mistakes over their relatively short time as a group, but what they did is, in this case, they simply went through the regulatory process, the governmental regulatory process, but did not really bother consulting the communities. So they come in then with a finding from the FDA that there's no obvious concern in terms of environmental impact, and then they just get ready to say, okay, we're going to go, and then the government says, well, wait, shouldn't, you tell us about it more and talk about it more? And then it got on the referendum, and then people were freaking out. And I think it's a really good point that you're bringing up, because people at that point were so afraid of Zika, and even in the face of their fear of Zika, they were more afraid of a GMO insect. So it highlights that issue. And one of the things that we're doing in California is we have a trans-UC collaboration that is aiming to try to come up with a gene drive system to combat the mosquitoes that carry Zika also, the Aedes mosquitoes. And our hope is, if we can succeed with the technology, to bring the technology forward that would be capable of doing that, is to bring it through our own regulatory process in the United States and in California, and at least show that we have the intent, if we could, to use it on ourselves in our own home state to diffuse what you rightfully point to as a potentially, colonialist-like attitude that would potentially be perceived that way. It's a very good point.
Thank you.
Yeah, no, I think just see, these larger issues of perception, I think, they change so quickly, right? It's very hard to kind of say that what's, in terms of optimism, momentum in the country. But I think on the politics, what I would say is both the major parties, right, have factions that are more modernistic and welcoming of technology, and have factions that are kind of, more traditional or just more suspicious of things. So, and my sense of all of this is that
the best way to have it played out is because our champions are kind of highly respected in the Indian public discourse. So whether it's the Tatas or whether it's, it's mainly the technocrats within the government, right? So the Secretary of Department of Biotechnology is a highly respected scientist. Samya Swaminathan, who's the ICMR head, is now WHO Deputy Director General. So the people who essentially supported getting through this entire TIGS architecture represent in some sense the pinnacle of public interest in Indian science, right? So to the extent that there has to be somebody taking a view on is this in the national interest, right? I mean, that's a view that they would take, and that's a view, those of us who are involved in the policy space will try to kind of provide the relevant information, but it's not necessarily our decision. So, I feel a little nervous about the extent to which we should be marketing versus we should be informing. I mean, this is a standard academic tension, right, between kind of dissemination, recommendation, and advocacy. And you see kind of academics kind of, jump over to the point where their objectivity as academics becomes questioned, right? I mean, and so in my own work, the typical, almost every paper of mine has substantial policy implications, but you'll just kind of disseminate in seminars and don't necessarily say, do this. And then, every 5 years or so, when there's a policy moment, I've synthesized a lot of my work on service delivery, say for the 12th five-year plan. So at that point, you go up to saying recommendation, saying, based on everything I've done, this is what I would recommend. Now, if you're an advisor within the government, that recommendation is heard a little bit more seriously. So now that I have this honorary kind of advisory role, at least they listen. Now, they're not like obligated to do anything, right? But in the end, it's a democratic, deliberative process. So, but I personally feel nervous about stepping over into what I consider an advocacy line, right? I mean, which is to say, saying we want to do this, and now we will, put on a PR blitz to kind of make it happen. That's a side of the line which I feel nervous crossing.
Well, I would agree with that totally. I mean, I think the decision as to whether to implement this kind of technology lies entirely within India and the decision-making process in India, and that all our role is to provide the scientific basis for whether it works or not.
So my question kind of relates to that. It's a little bit of a technical question. At the part where you talked about the eraser stage, is that motivated mostly to put people's minds at ease to say that, oh yes, we can cancel this, or is that something that is technically necessary? In other words, did you need that, or was that developed so that you can just say, well, we could erase it if we have to?
That's a great question. And, at one point I had my little 15 minutes in testifying before the National Academy of Sciences that eventually had a panel that looked at the regulation of gene drives and made recommendations. And my recommendation, which I was gratified to see that they voiced themselves in their report, was that these sort of reversal systems should not ever be used, if possible, and that the decision of whether to use or not a gene drive should be predicated simply on, do you believe the gene drive, in terms of its risk benefits, is got a positive benefit over risk ratio, and that the eraser type things would be developed partly as just part of the scientific entourage of tools that we use to study the process, and then have as a kind of last-ditch tool should something really go wrong. Because the hope would be is if you're going to really ethically release a gene drive, you should have no concerns of it being safe at that point. All the go, no-go decisions that get you to the point of where you say, okay, we should be able to release this, should have convinced you to the best possible level that one can have confidence in that it is safe. And then, if you're worried about things, which I find very difficult when people sometimes ask me this, is, well, how about the unknown unknowns? Like, well, what is it exactly that we want? But if there is such a thing as an unknown unknown, putting a second variable that you don't know about into the equation squares the unknown unknowns, and then that makes that problem even worse, whatever that problem is. And so I think that's the kind of concern I would have, is that really could create a weirdness, because we know from our own experiments that if you put an eraser in with a gene drive, there are certain kind of recombinogenic things that take place, and some of those are actually bad, like there are gene drives that don't have a marker on them anymore, so it's like not a good thing. Now we can engineer around that now because I think we know why it's working that way, but there could be other things that are complexities that we don't know, particularly at population levels. So my feeling, and I would hope that as things go forward, this would be the view that would be taken, is that these are kind of last-ditch effort elements if you ever use them. Mostly they're there for scientific analysis just to kind of figure out how things would work. And it's possible that under certain situations there could be, if you wanted to get near a border, there could be, if they were working well, there could be a decision that would say, what we'll do is we'll seed along the border the eraser as a protective zone, as a protective measure, and that would be something that, again, with a very large amount of, validating data, might be something that could potentially be useful in those situations. But it's a good question, and I would say no. In general, I would just go with the gene drive.
That's great, because you anticipated my second question, which is if you rely on that too much, people obviously will say, well, who is going to erase the eraser, right? But you basically answered my question.
I think, Elizabeth, you wanted to make a comment?
Well, actually, I had another question. So, there's a lot of excitement and a lot of interest in, the idea of creating a malaria vaccine. And, millions of dollars have been spent on it. And ultimately, in my opinion, the reason most malaria vaccine efforts failed was because of strain-specific immunity. So people did a lot of work in research laboratories in the United States, and they showed that this thing worked, it protected patients, but in most cases they were doing the challenge with their own laboratory strain of mosquito, or their own laboratory strain of parasites. And so they get really great data, and then as soon as they went out to the field and they encountered the huge amount of genetic diversity in the parasite, the vaccine no longer worked. And so that has killed a lot of malaria vaccine efforts. So I think we can get around this, but one of the things that you're going to have to think about is being able to test genetically diverse parasites and test them potentially with the vectors that they're going to be exposed to. And honestly, that's going to create regulatory headaches in just terms of getting parasites in and out, and just getting parasites from India that are native to test with mosquitoes that you've created here.
Oh,
so
right.
Are you thinking
No,
a lot
so-
about how you can,
Just on that last technical point, I mean, there's some very important other points that you made at the beginning, but the technical point, the idea is to have the mosquito work where there would be that test with the parasite challenge assays and so forth. Those mosquitoes would be built in India, and the strains of the parasites would be taken from India and tested in India. So there wouldn't be that. We're not trying to import any organisms. With regard to, would the immunizing mosquito vaccine fail in the same way that human ones might? I mean, that's a very good point. One of the antigens that Tony has selected is one that's completely invariant among all the parasites. So the hope is that that particular epitope would be pretty good. And the idea also of going in there with a combination of additional effectors. The other thing that Tony's very enthusiastic about, and some recent data looks very encouraging, is that one of the proteins that the parasite needs to interact with is a host protein called FREP1. And he has shown, and his subsequent work from people from his lab that have gone on with that, have shown that this protein is one that the mosquito can do without. And so the idea then is to take a guide RNA that would then destroy the FREP1 gene as the gene drive goes through the population. So that would be another level of reduction in parasite transmission. But you're right. I mean, until these things get tested in the field, we'll have no idea of what their efficacy is. And so one of the most important things, in my opinion, is just to get as quickly as possible to a point where we can just do the experiment. And that doesn't mean that we impose it on the people. I don't mean that at all. But just that we have the scientific information to know whether this kind of thing will work or not, and then provide that useful information to the public and to the government and to the deciding agencies, so that those kinds of decisions can be made rationally based on a better understanding of these kinds of important points. I mean, one of the things I just say parenthetically too is, in addition to kind of taking out things like FREP1, one of the other things we're very excited about, and Raja Kushwaha is back there, I got the 20th time that he's listened to me, is working on is, I think, a very exciting thing where you can also put potentially a guide RNA on the vector that sweeps through the population that cuts insecticide-resistant forms of certain genes, like genes that are ion channels to which the insecticides bind, but they don't destroy the normal functioning sensitive version. So what would happen, and we've got some data in the lab that's coming out that suggests this really does work, is you make a guide RNA that just cuts the resistant form but not the mutant form, and now as this thing sweeps through, it also sensitizes the mosquitoes again to insecticides, so that then again gives the standard vector control approach another element of leverage. So then I think that helps also kind of think about how it gets integrated with all the other things, including drugs, bed nets, and so forth, so that you have an enhanced overall eradication agenda, and that the gene drive is just one component of that hopefully strengthens the overall approach.
Any more questions? I think we are getting along in time, so maybe if there are no other questions, I'll just quickly ask one question, maybe from Elizabeth. So you mentioned that eradication or elimination of malaria was a goal. However, the approach that you have with drugs will only take you part of the way there. So do you have a sort of roadmap for how elimination could be achieved, and would it involve some kind of vaccine or active genetics or anything like that?
Yeah, I mean, there's what you could achieve with the existing drugs, and then there's also potentially what you could achieve with next generation drugs. And I don't think many of the models actually think about next generation drugs. So all of the economic models that I've seen, are based on the existing tools. But there's theoretical drugs that you could make that would just, prevent you from getting malaria in the first place, and then might be long-acting and might function just like a vaccine. So, that's kind of what we are aiming for is new theoretical drugs. And the other thing about the current existing therapies, and I think I forgot to mention that, but, you take chloroquine or you take artemisinin, and you feel great after it. You go back, you go to work, but you're still able to spread malaria around. And that's the real problem. This is the reason it persists, is because, it's just a total Band-Aid approach. And you've taken this drug, and then you go out, you go play soccer, whatever, and you're transmitting malaria to anyone that is bitten by the same mosquito that you're bitten by. And so you continue to go out and spread it. And so what we're really thinking about, in terms of next generation medicines, such as the spiroindolone that I talked to you about, is something that's going to prevent you from spreading malaria at the same time. And it's possible once we start putting these new medicines out there in the community that there's going to be a shift in terms of what's actually happened. And if you take out the transmission component, it's possible that elimination will start happening much more quickly.
Right. And I think, of course it's, propitious perhaps that we're both here looking at the kind of opposite ends of the spectrum of how to do it, but really in the end, the two together could be very complementary, because one of the things that, is even from that very simple cartoon that I first showed that's so obvious about malaria compared to, say, something like smallpox, is that it has life cycles both through the human and the mosquito. So it somewhat makes sense to try to take it out simultaneously in both, because those should be additive or cooperative activities.
All right. On that note, I'd like to thank the speakers again today for a wonderful discussion. And with that, we adjourn the meeting. We look forward to seeing you in more South Asia events. Thank you.